Hello. Oh. Justin's on his iPad today. That's good to see. Last time it was just iPhone. Michaela is here. Emmeline. Keegan Dupre. Brady Vassar. Hello, hello, hello. Okay, guys. I just thought I would just go over everything. Just go through the PowerPoints and talk about them. If that's okay by you guys. So we'll go ahead and get fired up. So, starting back at chapter 5, part 2, vertebrate development 3. I'm just organizing my screen here. I've got the chat open. If you have questions, you can chat there, or you can just talk. People don't seem to like to talk, but I end up doing all the damn talking around here. Okay, so I'm pulling up my notes too, just so I can see follow along with what we're doing here. Sydney. How do I make this smaller? Guess I don't. Okay. Shoot. Sorry, guys. I'm just kind of trying to organize here. Running out of real estate on my little tiny laptop screen here. It's frustrating. Justin, are you laughing at me? LOL means lots of love. Does that mean lots of love? Thank you, Justin. Lots of love back to you. Okay, so, oh shit. All right. I'll just make it up. I can't get the notes on screen. I'll just make it up as I go along. Did I say that out loud? Okay, so what we start out this chapter talking about, we ended the last exam talking about what's going on in that mesodermal region here between the node and where the differentiation happens. And that's the next slide. Oops, sorry. That's this whole bit here where you've got the node is releasing FGF, which is making Wnt, and that is producing a gradient. And as you get further away from the node, FGF drops, and you've got retinoic acid coming from the already differentiated part. So you've got those two gradients there, and you hit this one spot where FGF is low enough, is too low to maintain the mesenchymal phenotype. So then it switches, it differentiates, and it becomes a somite based on the RA influence. So that's where we left off. So today we're going to talk about, or in this part of the chapter, we talk about the rest of everything. So slide 2. Head structures. Head structures are all formed by when that node starts up. The first mesenchymal cells that enter go in, forward, and out, and they go underneath the ectoderm there, and they are going to induce the head structure to form. So the whole head has nothing to do with the node or the regression. It's just that mesoderm coming in and moving forward, and that's going to release some factors then that cause neurulation to happen there. And that's where your brain comes from. Okay. And now this is what we're on slide 3 now. Um, this is what happens in the node as it's regressing. So you've got regular old nodal cells, which are this tan line that the blue dots are on top of, and those cells come in and regress, and they become the nodal cord, and they're going to induce the cells above them to become neural plate. Now, at the leading edge of the node, you've got the stem zone, and the stem zone's producing more cells. They're neuromesendermal cells, so they could go either way. But the ones on top will regress with the node cells and become part of the nodal cord as well. And then the ones that go in and underneath those node cells become the somites. So this area here is where all of that is going on. That's the pre-mesodermal. Oh God, I can't remember the name of it now. That's okay. I'll make up a word later if I can't think of it. Okay, so, oops, shoot. So backing up to slide 3, that's what's going on here. You've got the cells are being made at the front of the node, coming around and diving in through because they're mesenchymal cells and they can move and they go in and form the somites and part of the nodal cord as well. Okay, so the next part with somatogenesis is the fact that these are patterned as they go through. And they kind of go through some experimental stuff here, talking about how if you flip that around, oops, no, they don't. First, we're going to talk about how the somites separate from each other. So your node's moving this way. You've got your differentiation wavefront behind it. You can see it right here. And you get separation of that solid strip of mesenderm into individual somites. And that happens because of notch signaling. You've got notch on one side and notch ligand on the other side. Notch ligands are serrated and delta. Usually we talk about delta. Pre-Semitic mesoderm. That's the word I was looking for. Pre-Semitic mesoderm. That's that area. Okay, so notch signaling then. Basically, notch is a receptor. It's a transmembrane protein stuck in the membrane here. And notch genes are repressed unless notch gets its ligand. So here's another cell activating notch. Here it's delta. And when delta hits notch, you get a proteolytic cleavage of the cytoplasmic domain of notch, which comes off and becomes a gene activator. So the nice thing about notch signaling is it's just cell to cell. It's not, you're not like releasing a hormone that diffuses off and hits a whole bunch of cells and does a lot of things. It's very short term or very spatially restricted. It only happens between these two cells. And that's why you get that nice separation between the somites. Okay, so here's where we talk about the patterning of the somites then. If you take a big chunk of the backbone or the back of the dorsal side here, flip it around, you get the somites have already been patterned by the time they form. So this experiment is showing you that as the somites are laid out, they're already patterned anterior to posterior, because you pull it out, flip it around, you still get that, you get like anterior, posterior, posterior, anterior, posterior. You get it flipped around backwards. And in the next lecture, we talk about why that is. So here we're talking about how many somites you actually get. And this is kind of important in that you get, this determines how many vertebrae the organism ends up with. And it's a function of both how fast the node is moving and this clock mode that's going on in the in that pre-Semitic mesoderm. So the node is releasing FGF, and let me see if I can find the slide here. The node is releasing FGF. FGF induces Wnt. Wnt is released. Wnt upregulates Notch, and then Notch upregulates Hairy, and then Ephrin A4 produces somites. So I imagine if you did this in a test tube, it would all just happen all at once. But here with this motion thing happening, you get FGF making Wnt, Wnt makes Notch, Wnt also makes Axon, which shuts Wnt off. So then Notch makes Hairy, but it also makes NSP2 and Lunatic Fringe, and that shuts Notch off. So basically you've got FGF is there, it activates all these downstream events, and these downstream events shut themselves off. And then once they've shut off, they kind of cycle back to responding to Wnt and starting over again. So you've got this cycle of activation and shutting off that happens, and that's our clock. And so I threw this slide in 13. I don't know if this is real or not, but it makes sense to me. So it's kind of a good kind of conceptual way of thinking about it. So FGF makes Wnt, Wnt makes Axon, and that whole process takes about 30 minutes. Axon then shuts off Wnt signaling, and then when Wnt signaling stops, then Axon stops being made. Axon eventually gets degraded, and then 30 minutes later, you're ready to respond to FGF and make Wnt again. So it's kind of this circular process that happens. And the speed of that depends on what organism you're in. In chicks, it's about 90 minutes. In mouse, I think it's like 2 hours or something. So the speed of that, again, determines how many somites you get, because the node is moving at a constant rate, and every time it cycles through, it will leave a somite behind. So the faster it cycles, the more somites you get, and the smaller somites. What else? Okay, I think that's all we had to say here. So just backing up to say it one more time, here on slide 11, you can see the node and the presemitic mesoderm as it's moving in this direction. And as this gets far enough away that the FGF drops off, then you stop the cycling, right? Because FGF is starting the cycling. Once you get a decrease in the FGF concentration, cycling stops, Notch is upregulated, you get your somite. Okay. Going back to AP patterning here again, if you take a chunk of some very posterior somite and put it more anteriorly, you get a pretty messed up bird. Okay, so I think this is illegal in some states. Okay, so and the reason for that is those somites are patterned as they're laid down. And the reason for that is because of these Hox genes. So you've got 4 different clusters of Hox genes, and they're paralogous, which means Hox A, you've got a gene 1, B, you've got a gene 1, D, you've got a gene 1. These are all paralogous going up and down this way. And these will be expressed from anterior to posterior and in that order. So in the anterior regions, the first ones that get turned on will be your HOX1 genes. All A, B, and D will get turned on, and the HOX1 genes will then turn on the later genes, so they get progressively expressed as the node goes down. And these Hox genes, here's paralogous. You can see how the expression starts at the anterior part in the basically the base of the skull, and some of the face things will be a somite, and then from there it goes down the spinal cord all the way to the tail. And this is showing that the beginning of where a Hox gene starts to become expressed is very sharp, but then it tails off. So when you get towards to the tail, all the Hox genes are on, but the first ones are very, very low in concentration. So the front edge of Hox expression is very sharp, but the back edge isn't, and they stay on pretty much all the time, except at low levels. So then each somite is then patterned by which Hox genes were activated when that somite was laid down. This is slide 19. So here in the chick, we've got all these cervical vertebrae, some thoracic, some lumbar, and then some tail vertebrae here. And if we compare that to mouse, we've got the same Hox genes in cervical vertebrae. The difference is you've got more cervical vertebrae in the chick. But if you look, here's C5 at the end of the cervical vertebrae, here's C5 at the end of the cervical vertebrae. So the patterning is the same across species. It's just that the number of somites will change, which will then change the length of the spine. Hoxerator. So keep bringing this up. So again, you flip that over. What you're doing is flipping the Hox expression gene, and so they end up backwards. All right. So Christine, are you still there? She didn't like this last time I played it. I'm going to play it again. Okay. So here I'm trying to show how that Hox gene expression gets patterned into a somite. So you've got your node, your pre-mesodermic or pre-Semitic mesoderm moving in this direction, or the wave of that moving in this direction. The cells are staying there. But so as that node and pre-Semitic mesoderm is moving in one direction, you get a wave of activation of Hox genes going past it, wave after wave of Hox genes going past. And whatever Hox genes are on when cells pass the differentiation wave front, those are the Hox genes that are expressed in that somite. So if we throw this up, you can see here comes Hox gene 1. The wave of its expression moves down, and the cells that are the somites that are being laid down are being patterned by that Hox expression. And I kind of shortcut it there and bring the node all the way to the end to show that the whole thing gets patterned that way. Okay, Christina, does that make sense now? Christina, yes, okay. You took some notes, okay. All right, so that's how you're getting your AP patterning, your large scale AP patterning. Here's why they get turned on in that sequential way. Your Wnt signal, which is coming from the node, is activating the first Hox genes. And the Hox genes also, or the Wnt also activates CDX2, which activates the later Hox genes. So now you've got later Hox genes going. So the earlier ones turn on the later ones until you get all the way to the last one, Hox13, and Hox13 shuts off the other ones. So then it shuts off when you get to the end of it. Wave of gene expression. Okay, so now we look at patterning inside each individual somite. So you've got the overall anterior posterior patterning, and within each somite, you've got an anterior and a posterior side. And that's set up by notch signaling. Notch and delta come together to activate MSP2, and MSP2 blocks notch. We saw that earlier with the clock situation. So what happens through this is you get delta and MSP are expressed in non-overlapping bands within a somite. So you've got delta at one side. This is a somite, a stripe of somite. So on one side you've got MSP, and at the other side you've got delta. So within each somite you've got that AP patterning. More interesting, in my opinion, is the dorsal ventral patterning and the midline to lateral, medial lateral patterning. So here's your somite. This guy here is covered by epidermis, of course. Dorsal up here, ventral down here. Here's the notochord. So this somite then differentiates into sclerotome and dermomyotome. So sclerotome will become skeleton, basically cartilage, and then ribs and spine. What do you call spine? Vertebrae. Vertebrae. How's that for anatomy and physiology? I never had anatomy. Okay, so here's your dermal myotome, and that will then differentiate into basically dermis and also muscles. The muscle cells will migrate. So they undergo that EMT transition. So the medial myotome over here is going to be different from the lateral myotome. The medial will be axial muscles going up and down your back, and the lateral will be limb and side muscle. And then this dermis then is the layer underneath your epidermis, so it's skin. Sclerotome, again, we're being migratory here, and they start to migrate. They go around the notochord and then form the vertebrae. If you remember way back to like chapter 3, we said, what does a notochord become in the end? All that's left of the notochord in the end is the little spongy discs between the vertebrae. So you can see here that the sclerotome is surrounding that, and then they end up squished in the middle there. More importantly, why is this happening? If we move on to slide 29, you can see we've got 3 different gradients here of hormones that are causing this to happen. Up here, the top of the notochord is releasing Wnt. So Wnt is inducing the you've got a dorsal ventral gradient of Wnt, which is causing the dermal myotome to differentiate on top, and the lack of Wnt is giving you the sclerotome down here. And then from the bottom up, you've got sonic hedgehog. So sonic hedgehog is causing the sclerotome to form down here and not up there, because you're getting it down here. And then medial to lateral, you've got BMP4, which is coming from the mesoderm on the sides. So that gives you the difference between the middle back muscles versus the limb and side muscles. So you've got 3 different gradients there that are patterning the dorsal ventral and medial lateral aspects of the somites there. Okay, Sonic Hedgehog. So this is the first time we've seen Mr. Sonic Hedgehog. This is a figure I got from a different book. I didn't like the one in this chapter of your book. There's a better one later in your book, but it was too late because I already had this one in here. So basically, for hedgehog signaling, smoothend is in a vesicle underneath that subcellular pool of vesicles I'm always talking about. That's where smoothend is, and patch is blocking that from fusing with the membrane and coming up. And inside this primary cilium, which is a cilium that doesn't move, it's just a different type of cilium, you've got this protein GLI. And if smoothend isn't there, GLI will get cleaved, and the released part of GLI then becomes a transcription factor that is repressing transcription. So it's a transcriptional repressor. Nora? Nora? Can't get any help from the kids anymore. They've given up on me. Okay, so, but if hedgehog is around, then hedgehog will bind to patched. It moves away, so it's no longer blocking smoothend. Smoothend fuses with the membrane, goes up into that primary cilium, and it causes GLI to be released without being cleaved. And then it's a transcriptional activator instead of inducer. So it's kind of a weird signal transduction mechanism. But the second one we saw today where proteolytic cleavage was part of it, which is kind of interesting. Boy, the things cell biologists find interesting, I don't know. All right, so now the next lecture was talking about neural plate development. So first we just look at the overall shapes of things changing, and then we talk about the induction by different hormones. So neural plate starts out being induced from epidermis by the notochord. So the notochord's making epidermis in the neural plate. And the neural plate, oh, we see this again later in chapter 7. Why don't we throw some of that in here now? be kind of sussy, wouldn't it? So here's your neural plate, and now you get these cells at the bottom that bend, right? Change in cell shape. You go from columnar into like a wedge shape. So the ones at the bottom become wedge shaped, and that causes the neural folds to come up. And then you get more cells bending in the center here, one on either side, and that brings the sides together, and then they fuse. And they're fusing, again, reviewing from chapter 7, fusing because they're putting out the right cell adhesion molecules, and they're binding to each other and releasing cell adhesion to the ones above them, the epidermis. So anyway, your neural folds fuse into a neural tube, and the dorsal side of the neural tube becomes neural crest cells, which undergo epithelial to mesenchymal transition, again, EMT, and crawl off and go off into every which way, and make all kinds of beautiful things. And then the epidermis is closed over the top of this whole structure. Okay, why is this happening? So I think I know I told you to watch this video. This woman in Barcelona did this, and she did a really nice job on it. So here is the beginning part. You've got notochord here with neuroectoderm on top and non-neural ectoderm next to it. So this is going on because you've got Wnt signaling here that's converting this epidermis or ectoderm into neuroectoderm. You've got BMP4 over here that's inhibiting neuroectoderm. And right in the middle, you've got an area that's got some Wnt and some BMP4, and is kind of on that transition area. So that ends up a little different, and that's going to be your, eventually be your neural crest cells. So then you get, the cells bending down here and coming out and closing. And then once it closes, these cells in the neural crest are now exposed to a lot more BMP4, and that's causing them to become, they're upregulating snail, twist, and another transcription factor that slips my mind right now. Let me see if I can pull that up. I'm guessing we are in slug. Right, thank you, Nick. Slug. So then because you've got those transcription factors upregulating, you get that EMT again, and then they sneak off and become a lot of things. And it's amazing how many things they come. Here we've got slide 37. You get glial cells, both types of glial cells there, melanocytes, which is kind of weird, goes everywhere into your skin to give your skin color. Sensory neurons, slug. I think SOX 9 and 10 cause snail, right? Then once the tube closes, slug and twist. You're correct. You're right, Nick. So you get your sympathetic and your parasympathetic neurons, your neurons that are regulating your GI tract, and some cartilage there. So they go off and make an awful lot of different things. Okay, those are your neural crest cells. Tissue that the cells differentiate into is dependent on their origin on APA. Okay, so this is the kind of a little bit complicated, but not really, how like these neural crest cells are like multipotent, because they go off, they can become a lot of different things. And if you take them out of where they are and put them in a different place, they'll become something else. So they're multipotent, but they're not pluripotent, because they do have specific Hox genes activated based on what somite area they originally came from, right? So they're not totally pluripotent, but they are multipotent. They can become all these different tissues that we talked about a minute ago. But they're always going laterally. They're not going anterior or posteriorly. They're just going laterally, and they're sticking with that Hox gene specification. And then here at the end of chapter 5, we're talking about this is talking about how the Hox genes are still being represented here. So here we're talking about how these rhombomeres, which are actually, I guess they're kind of analogous to somites, except they're coming from the neural tube. So you've got these transiently delineated sections of the neural tube. Hang on a second, the dog needs to go up. You're a coward, Lorraine. Okay, lay down. So the neural crest cells that come from a specific rhomboid will make a specific structure. So here you're making jaw, over here you're making gill arches or whatever. Here, this is better. So you're getting throat here from R4 and R2. So now you can take these stem cells and move them in the posterior direction, and you'll get the same tissue you would have had if they stayed in their particular direction, particular spot. So in the next slide here, we take R2 and we put it in the R4 space, and it still makes jaw. So now instead of getting a throat, we get two jaws, which makes for a very bad kindergarten pictures. Ugly kids. And then the last thing is left in this chapter is left, right. So here we're looking at what's going on in the node itself as it's moving forward. So as that cell is moving forward, you've got cilia in the middle of the node that are swishing in one direction. It's like this. And you've got cilia on either end, which are not modal cilia, but they're sensory cilia. So they're sensing this fluid flow, and the fluid flow is activating these sensory cilia, which then causes calcium release in those cells over there. And then the book says, I don't know what happens then, but something happens. Basically, it says they don't know if it's the calcium that does it or the fluid flow, but for some reason you're getting more nodal on the left-hand side. So is it because the nodal from the right's being swished over to the left, or is it because the calcium, interior calcium being released causes production of more nodal? Stay tuned. 10 years we'll know. Okay, but basically that's mouse, and mouse is pretty simple compared to chick. So the next one is chick. So here in chick, you've got hydrogen potassium ATPase in the node, and it's higher on one side than the other side. So you've got more hydrogen potassium ATPase on the right side, I believe. Yeah, and that makes an electric potential between right and left. Now, they haven't said why you get more hydrogen potassium ATPase on the right side. They just said you do. So we still don't know why there's left-right, basically. But anyway, you're getting more hydrogen potassium ATPase on the right side, which is affecting the electrical potential on the left side, which is causing an increase in extracellular calcium. Now, calcium activates Notch signaling by increasing Notch ligand. And Notch signaling then together with sonic hedgehog signaling. So your node, back up one, just so I don't have to BS this. Okay, so you're getting Notch signaling because of the electrochemical potential there. And the node is also putting out sonic hedgehog. So the combination of Notch and sonic hedgehog on the left is causing nodal production. And on the right, you've got activin, which is blocking sonic hedgehog. And then you've also got lefty on the right, and lefty blocks sonic hedgehog as well. No, it blocks nodal, I'm sorry. Lefty is being produced on the right, which is also blocking nodal. And so bottom line, nodal's left, non-nodal is right. And if you don't have any of this signaling, it's a 50-50 which way it's gonna go. You can have situs inversus if you don't have this. And again, with this figure, I'm always confused because they put lefty on the left, but its effect is on the right. So I don't understand why that is, but there it is. Okay. Questions about chapter 5 before we move on to chapter 7? Can I get an amen from the crowd here? I'm getting some ups. I got an Emily, a Sydney. Rachel Siller, give me a thumbs up, thumbs down. Oh, there's no thumbs down. Julia Waters. Justin, are you awake, Justin? Jesus Christ. Okay, there you are. Sorry. Sorry, my bad. My bad. I'm not sorry. You know I'm not sorry. Okay. Michaela's good. Okay. All right, so that's chapter 5. By the way, I checked I put a quiz up. I don't know if you guys noticed. Okay. I put a quiz up. I don't know if you guys noticed, but only one person took it so far. So check out that quiz. There's some good questions there. Let's do a slide share on PowerPoint 7. Dun, 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 dun. Now here we're less, in chapter 7, we're less worried about hormones than we are about connections between cells and contractions of cells, changes in cell type. So I'm going to get rid of my notes because I'm not looking at them anyway. Sorry, I'm just re-establishing my real estate here. Okay. Chapter 7, morphogenesis, or change the production of shape, morphogenesis. Okay, so morphogenesis, this whole chapter is all about 3 or 4 different things. It's about cadherins, integrins, contractions of cells, and cells migrating, so 4 different things. Cadherins are cell-cell connections, and they're kind of keeping tissues together. Integrin, and here you can see that, so here's your cadherin, and adherens junctions are the most important ones, the ones we talk about most, and they are between the same cadherin. So 2 cells expressing the same cadherin will attach to each other. That's kind of important because when we start separating cells, then the cells stop expressing the same cadherins, and now they can't stick to each other, and they can move with respect to each other. So typically when we're doing an EMT or an MET transition, it has to do with cadherins. Here's adherens junction. So remember, these always involve some kind of signal transduction. So I don't know who's here from cell biology, but I asked a question on the last cell biology test about adherens junction and integrins. How do they signal into the cell, and how does the cell signal back out? Everyone got this side, but they didn't get the signaling out part. So I'll talk about that more here. Basically, when cadherins bind to other cadherins, that activates some of these signal transduction molecules that attach them to microfilaments. These are actin microfilaments. So that connection between the cytoplasmic side of the cadherin and the microfilament involves some structural proteins, and it also involves some signal transduction proteins. So when they attach, they activate signal transduction, so the cell knows it's attached to something, attached to another cell, I mean, specifically, because these are cadherins. Recognition goes in the other direction as well. When the cell wants to let go, it signals that to the cadherin, and on the other side, the cadherin lets go, so the cell can separate then from other cells. So that's what I mean by signal transduction, going in the opposite direction. The cell can signal the cadherin to let go. Desmosomes, let's not worry about these. These don't come up again, but basically desmosomes are kind of like adherens junctions. The big difference is that it's between 2 different cadherins. So it's not E cadherin to E cadherin, but it's 2 different cadherins attaching to each other. And instead of attaching to microfilaments, they attach to intermediary filaments. But this isn't that important. It doesn't come up again, so I'm not going to put it on the test. Don't worry about desmosomes. You could write home and tell your mother you learned about it, but other than that, let's not be concerned. Okay, this is important, integrins. Integrins are attaching to fibronectin, I think collagen too, but extracellular matrix in general. So, and this is keeping the cell together, attaching it to that extracellular matrix. And again, let's see, these are gathered together into focal adhesions. And you've got your structural proteins and you got your signal transduction molecules out there. Oh, excuse me. So when these are attached, the cell knows that it's attached to extracellular matrix. And also, signal transduction going the other direction, the cell can say, okay, integrins, let go, and they'll let go. And then that allows the cell to walk along the extracellular matrix. And that's going to be important for cell migration or cell crawling, the ability to regulate those connections. Okay, so now I go into cytoskeleton a bit for movement and contraction and changes of cells. Microtubules are involved in a lot of things. And microtubules, whenever you have microtubule-based movement, it has to do with these motor maps that are kind of like walking along the microtubules. That's all. That's the only reason I put this out here is because the chapter talks about motor maps, and so there's your motor maps. Okay, so here's your cilia, contract because of a motor map, or not contract, flap back and forth, let's say. Microfilaments then are based on actin, and you've got different structural arrangements of actins. Here is antiparallel sheets that will cause the cell to contract. So when it's crawling, that pulls the body forward, and then integrins grab further out, pull the body forward, grab further out, pull it forward. So that's what those things are doing. And out here is the kind of, the lamellipodium is where it's reaching out towards to make new focal adhesion connections to move in that direction. There's your lamellipodium used in cell crawling. It's a beautiful thing, people. Okay, adherens junctions. So this is the next, is this the next? I don't know. I think this is the next lecture, but I'm not sure. So adherens junctions then are help to sort cells out. I guess that's what we're trying to say here. Cells with the same cadherins will bind to each other. Those with different won't. So here they're showing that in an experiment where they took some mesoderm and some ectoderm. They dispersed each into individual cells, put them together in the same tissue, and the mesodermal cells grabbed each other, the ectodermal cells grabbed each other, and they formed two separate tissues. So that's just, again, showing that cells will sort themselves based on what cadherins are available or are being expressed in me. Another important thing for cadherins are when they let go. So here on this slide, which is slide number 17, you can see that EMT, epithelial to mesenchymal transition, where those red dots are cadherens junctions or adherens junctions that are holding cells together. When they let go, the cells are able to slide away. They'll change shape and then start crawling away. And they may still be slightly connected. We talked a little bit about these gap junction proteins that are allowing them to communicate with each other. But for the most part, they're not very organized. They're just sliding away versus epithelial cells, which are very organized and stuck together in a tissue. Okay, compaction. So here we'll see how making these adherens junctions then is kind of sorting the cells out, but also activating hippo signal transduction. I'm sorry. That's all I only have one slide on that. I talk a lot about it in the notes, but I only have one slide. So basically, when you go from the 18-cell morula to the 16-cell compacted morula, cells that have got adherens junctions all around them get shifted to the center of the embryo, and ones that have a free side, a free apical side, stay on the outside. And that free apical side doesn't allow hippo signaling to happen. The covered on all sides activates hippo, and that activates gene transcription. So the CDX2 is going to be expressed on the outside and OX4 on the inside. And that's because of something we talk about a lot in this chapter, which is surface tension. When this thing is attached at all sides, you've got actin myosin kind of contracting on the surface and putting tension on the surface. And so, let me shut this off. So that's surface tension and its import. All right, so now we're going to talk about convergence and extension and how you go. I think the best picture is this one. This is slide 23, where you start out with a band of mesoderm all the way around the embryo, and this band gets sucked up through a hole, and it turns from a band into a sheet, because you need a sheet to make an embryo when you go from radial symmetry to 3 axes of symmetry. Okay, so what is convergence and extension? Basically, cells aren't dividing here. They're just intercalating and coming closer and stretching out. So it's just like I said here, like you take that cylinder of clay, you put it on the table, you do this, and it rolls, and it pushes itself out. And they're involuting through this blastopore, which is a small hole compared to the size of the embryo. So they all have to squeeze through this little hole. So when they get there, it's kind of like you're trying to get into a concert and there isn't a line, everyone's crowded at the front door. And so as you go through that door, you intercalate and then you make a line going through. That was a great metaphor, Dr. Slish. I just came up with that. Yeah. Or you're getting off the north way where it's a you've got like all these lanes coming together, construction on the highway, right? All these lanes have to merge into one lane, and when you get to the front, the cars are intercalating, unless you're in New York and people just cut each other off. All right, so here's intercalation. Slide 22, you can see you've got this fat tissue, short fat tissue, and the cells intercalate and they converge and they extend. Convergence and extension. Okay, and so here we are back again at this slide where you can see all these cells are being sucked up through that little hole and they're making a sheet. And when they do that, oh, this is a beautiful picture down here too. This is brachyury, which you know is a mesoderm-specific transcription factor. So you can see it's a circle here in this mesoderm, and then it all gets sucked through the blastopore and it becomes a line. Convergence and extension. And then this is an interesting slide, slide 24, which is talking about how cells that are near each other in the blastula could be far away in the gastrula, or things that are far away end up close together in the gastrula. So things move around a lot during this involution. All right. So here we're talking about convergence and extension in the chick gastrula. So the formation of that primitive streak is all about C&E because cells are coming from the side, starting at Kohler's sickle there, coming from the side and coming in like this and moving through, and that's what's forming that primitive streak. It's mediolateral intercalation. Oh, did we talk about, hang on a second. Record, bada bing, bada boom, bada bing. One thing I forgot to talk about, you forgot to talk about, I'll talk about it, is epibole, which is also convergence and extension. It's radial convergence and extension because you've got like when you're at the blastula stage, the ectoderm is all at the top. You know, you've got kind of a thick layer of ectoderm that then thins out and spreads around the cell. So that epibole isn't because of cell division so much as because you're converging and extending around. So you've got your medial lateral and you've got your radial convergence and extension. Okay, primitive streak. Okay, so here we are converging and extending on the primitive streak. So you've got apical constriction here. So again, actin myosin is pulling these cells in to converge them. And then once they get to the primitive streak, you get an EMT conversion. So the cells that get here let go of their adherens junctions. they become mesenchymal, and then they not involute. What was the word we used for that? Ingress. So they ingress and then skitter off in their own directions. And here's another example of convergence and extension with a notochord. So if you ever watch the the actual time lapse of a gastrula being formed, you notice how it just stretches. It starts out as a ball. You see you kind of see the activity of the gastrulation, and then you see the whole thing go just kind of get long. And when that happens, it's because part of it is because your notochord there is going from this fat little patch of notochord into a long extended form. And it's because the cells are changing shape. They're intercalating, coming across each other. And here's another shape change. They look like that. And then they keep intercalating and changing shape until you get a cord out of it. Pizza-shaped cells. Okay. Oh, and this I talked about earlier when we did neurulation before. This is cell changes, again, because of apical constriction. So this is neurulation. Here's your neural plate cells. The ones at the very center undergo apical constriction. So the top part of the cells gets small, the bottom stays big, so you get a wedge-shaped cell. That brings the sides up. So now you've got this neural groove. Once you've got a neural groove, you get a similar thing happening here at the dorsolateral side. So that brings it, so you go from this to this to this. And now the two ends can touch. And once they do, the neuroectoderm lets go of the surface ectoderm and grabs onto other neuroectoderm, and you close that neurotube up. Here's these protrusions of lamellipodia in the surface ectoderm that grab each other and then close up the surface ectoderm, and then they let go of the neuroectoderm, and then you end up with a little space there. Fusion. Here's what happens if it doesn't fuse. Let's not look at that too much. And here we are summing up the 4 different movements that we talk about in this chapter. You've got migration, cell crawling, which involves lamellipodium extending, focal adhesion making contact, contraction of the actin myosin stress fibers pulling the cell forward, and then the integrin letting go at the back end. And it doesn't really happen, it's constant, it's constantly happening, but those are the 4 things that happen. And then you've got convergence and extension with intercalation. And you can see there the lateral callation versus the radial callation, 2 different types that we talked about. And then ingression, which involves cadherin or adherens junctions letting go, and the cells undergoing that EMT transition. And then shape changes, which we saw in the notochord, and we saw in the neural plate. Apical constriction, changing the shape of a cell, and then because all the cells are connected to each other, that changes the shape of the tissue. Woo, Nelly. Boy, it's been a fun semester, guys. Sort of. I miss seeing you guys, though. Let's see if we can't bring everybody up here. That's not going to work. Okay. Rachel Cilia's here. Okay. I guess that's all I got. There's Nick. Hey, Nick. Did you get a haircut? Where'd you get a haircut?
Yeah, I told you I shaved my head.
Did you? Cool.
Shaved my head. Yeah.
I should do that. It's getting so out of control here. I do have clippers.
Yeah, I tried to get my mom to cut my hair, but she did a really bad job. So I just said shave it after that.
There you go. There you go. I'm trying to get my wife to do it, but she keeps saying no. Ah, there's Christina.
Hi.
Where are you, Christina?
I am in my house in my kitchen.
In Montreal?
Yes, in Montreal.
So this is an international Zoom we're doing. Woo!
So
Rachel's not wearing her hat today. That's nice. Emily, where do you go? Emily, are you going to Buffalo or Albany after this?
I'm going to Buffalo.
Buffalo.
Yeah.
Cool. Sounds good.
Yeah.
There's Michaela. Hey, Michaela. She's not talking. That's all right. I don't take it personally. The rest of you want to show your faces? Honestly, I'm looking at the names of people who haven't shown their faces. It's okay, guys. You don't have to show your faces. All right. Well, that's about all I have. Exam in 2 days. I haven't written it yet. Look at the practice quizzes I have out there. It'll be similar to that. Any questions, Adya?
Lish, I sent you an email.
Yeah, I don't think so. Can you send that again? If I haven't answered it, maybe I didn't get it. I decided to do it later.
It was only just before 2 o'clock.
Oh, okay. No, I haven't been on since then.
Okay.
Okay. Any questions or comments? Nothing? Nope. Sydney says nope. All right. Call it a day. I will post this up if you want to see it again. I don't want to see it again, that's for sure. All right. Well, I'm going to stop. Okay. Everybody ready? Oops. Stopping.